Okay, this is for day 2. These are going to be short answer notes. Your short answer section actually isn't very long today, or long this time, but you're going to have to do a few matching on this one, as well as write out short answer answers. Matching cell structures to functions. So cell wall, of course, it's for support. Nucleus, it's the control center. Smoothie R is for modifying and sending lipids. Ribosomes make proteins, and mitochondria oxidative phosphorylation, or the powerhouse of the cell. Protein synthesis, match the molecule with its job. tRNA is what goes and gets the amino acids. mRNA is the code that it reads. rRNA, it makes up part of the ribosomal complex. The codon are the set of 3 bases that are on the mRNA that it codes for the amino acids, and the anticodon are the complements of those, and those are found on the tRNA. Biological molecules, you're going to match the molecule with its description. I don't off the top of my head remember, give me just a second, what that is. So let me pull up the exam and scroll up to see if I can remember what those are. Okay, so you have to know that carbohydrates, an example of carbohydrate is cellulose. All of them have a carbon skeleton or have carbon as part of them. Lipids are fatty acids or have fatty acids inside of them. The nucleic acids make up your genetic material, and proteins have peptide bonds in between all the amino acids. Match the organelle with the type of cell it is associated with. So if you're watching this, you get a little helper here. Ribosome, central vacuole, endoplasmic reticulum, and mitochondria are all of the organelles. So ribosomes, of course, are found in all of them. Central vacuole is only found in plant cells. Mitochondria are found in plant and animal cells. And the endoplasmic reticulum is found only in eukaryotic cells. Match the type of membrane transport with its description. Of course, simple diffusion is just where molecules move from a high concentration to a low concentration until equilibrium is reached. It does not require energy. Facilitated diffusion is that same process, only instead of it just doing it, it has to have a protein that goes through the membrane for it to pass through. Glucose goes through facilitated diffusion. Active transport requires energy for it to take place, and oftentimes it does require a membrane protein for that to happen. Not always, but often. Osmosis is where water uses a membrane protein to travel through the membrane from high to low concentration, not requiring energy. And facilitated diffusion and active transport are 2 together, so they might be talking about both of those. You can't say use energy, but you can say they both use a protein in the cell membrane. So you'll have to match those with whatever descriptions they have. Then you have only 7 short answers. It's actually a very, very short section this time. So it says, given the sequence of DNA along strand A, there will be a strand of DNA going across your paper. Write the sequence of bases that would be found in the complementary strand B. So of course, A pairs with T, G pairs with C, and you'll have to just write those out. Then it says, assuming strand A is the template strand, the strand that is transcribed, meaning left to right, write the sequence of bases that would be observed in the resulting mRNA molecule. So again, you're going to take that strand A. Let's say strand A is A, T, C, G, A. And its DNA complement would then be T, A, G, C, G. But if you're doing your mRNA off of that, A pairs with U, T pairs with A, C, G, G, C, A, U. So this would be your mRNA that complements. This is strand A, this is strand B. Pretty pen, give me all kinds of sparkles. Letter C, write the sequence of amino acids that would be linked together when translation is complete. So this is the activity you did with Mr. Johnson. Oops, undo. We don't use the TAG. UAG, I think it's actually a stop codon, so this wouldn't get us anywhere because our thing just automatically stopped. It should have been AUG if I wanted to make meth, because everything starts with meth, and that's where we always begin, but I messed that up for my example. Wasn't thinking ahead. But every 3, you would use the chart or the circle. I'll have both of them on your test, and you will match them to those amino acids, so you write those out. What happens if there's a mutation in the DNA sequence? Well, maybe it does nothing, because it might not mutate the amino acid, or it mutates either one or very many mutated amino acids, depending on what happens. So you've got to talk about how more than one thing can occur there. What is the term differential gene expression? That comes from this time's notes. Basically, genes differentiate as they develop in an embryo. So it means that certain genes will be turned on in certain cells and turned off in others. Like all the genes that are possible to make your cells anything, your beginning stem cells, they're all turned on at the beginning because your cells can become anything. But then as they develop into the line where they're going to become the integumentary system, or where they're going to become muscle cells, other genes will turn off and only leave some on. So your stem cells like become less specific at that point, that can make those types of cells, and then they become even more and more and more specific. So differential gene expression is where your genes are only expressed as they are needed to differentiate for those types of cells. In terms of cellular respiration, why is it necessary to incorporate plants into a terrarium, a sealable glass container containing soil and plants, along with fauna, which are animals? Well, think about it. The waste products of photosynthesis are what's needed for cellular respiration, and the waste products of cellular respiration is what's needed for photosynthesis. So in order to recycle those molecules, you have to have both plants and animals present. Otherwise, you will use up all your oxygen. Or if you have plants, you will use up all the carbon dioxide, and nothing will be adding back into the system and recycling your molecules. Explain how the equations for photosynthesis and cellular respiration compare. You just need to say that they are basically reflections of each other, with the exception of energy, because energy in the end of cellular respiration is ATP, and at the beginning of photosynthesis is light energy. What are transcription factors, and how do they relate to gene expression? Transcription factors are different proteins and RNA pieces that can attach to DNA during transcription, which is when you're making your mRNA, that can either cause the transcription to speed up, or cause it to slow down, or cause it to stop, or cause it to begin. So transcription factors are just different pieces of molecules, different molecules that attach to DNA to signal, hey, we need this gene now, start transcribing, or whoa, we don't need you anymore, stop. So there's all kinds of transcription factors that you'll learn about in our notes this upcoming week, or next week as it seems to be. Given a scientific experiment scenario, determine the following. Again, independent variable is what you are going to change. Dependent variable is what you are going to measure. And two constants are things that you will keep the same in every piece of the puzzle. So like the amount of fluids, or the amount of soil, or the amount of light, or whatever, those are your constants. And then given a scientific experiment with data, be able to write a conclusion statement. So what is the claim, or what conclusion did this person come up with? Guys, in AP Bio, this is your CERR. Just keep it short and sweet. They're only 2 points apiece. But what conclusion statement did it have? We just did our lab paper, so you should be good at this. Write a kind of a statement for the results section. So support it with evidence. Give me the numbers part of it. Give me a quick discussion. Talk about the science of why it happened. Scientifically back up your data. Don't just say, yeah, it happened and that was good. This was a great experiment of osmosis. This experiment of osmosis is great. Don't do that. But just tell me why it was good. Explain it, and then validate it. I guess that should have gone with validate, but validate it. Tell me, were there any flaws? Is it a solid experiment, or was it crappy? Did they not have a good control, or whatever it is? So you're going to do a little bit of a lab write-up at the very end, because that's what we've been working on all year, and can you do it on the fly? So that's your short answer notes, and I hope those are helpful. Yay, final exam!